All right, guys. We're going to talk about some very interesting topics. It has absolutely huge potential, but there's also, wherever there's high potential, there's also significant risk. So we're going to talk about both these subjects. And frankly, what it comes down to is what makes us different. We all sort of think we're all different in some way or another, but what makes us different? Well, I wanted to pick a human being to talk about first. This is Albert Einstein, actually, in case any of you noticed, although he's in beach clothes on the beach. Okay. He weighed about 150 pounds. And what's interesting about this is, what was he made of? He was made of 97 pounds, roughly, of oxygen, 27 pounds of carbon, 14 pounds of hydrogen, 4 pounds of nitrogen, and a whole bunch of other smaller things. But the fact was, if you reduced him to his atoms, he was that. Now, what I'm going to do is I'm going to show you something pretty amazing. A 150-pound Great Dane reduces to the exact same numbers. It isn't what we're made of that makes us different. It's how it's organized. Mammals are all the same stuff. By mass, because oxygen is heavy, it is the single biggest part of our bodies. But on the other hand, by number of atoms, hydrogen is the biggest one. And you can see from that hydrogen, the green and the blue, we're mostly water, is what it comes down to. But the fact is, almost all mammals are exactly the same thing. Land mammals are the same thing, okay? So it isn't what we're made of that makes us different. It's how they're organized. It's what it's attached to what things are bonded to what, how they pass around energy, for example. And this is all information. We are all big piles of big data, is what it comes down to. That's what makes us different, is the way we're organized. We have approximately 100 trillion cells in our bodies. And each one of those things has 3 billion base pairs in it. So you can immediately realize we're talking about huge amounts of information inside our body. And then we've got epigenomes, we've got microbiomes, we've got immunicomes, we've got viromes, communicomes. We've got a whole bunch of different proteins inside our bodies that are different, and they are all in huge numbers. And furthermore, they change over time. The number of cells in our body obviously grows as we mature. When you're a little baby, you don't have 100 trillion cells, but when you are an adult, you have 100 trillion cells. Our genome is pretty stable. It does change a little bit over time, but it is pretty stable. But all these other things are changing, and in some cases they're inheritable. Epigenomes are inheritable as well. So medicine is becoming big data, effectively. And doctors are becoming data scientists, effectively. It's all code. It's just quaternary instead of binary. Now, historically, we have, over the last few millennia, been steering evolution. If you look at what corn looked like 2,000 years ago, it doesn't look like corn today. We have been selectively breeding. So we have been, in fact, already modifying the genome some. Okay? But we've been doing it by selectively mating things to other things. Frankly, pets, just think about dogs. Dogs all looked the same 2,000 years ago. Now there are so many different breeds, from Great Danes to Chihuahuas. So it's changed a lot because we've been interbreeding things and making things happen. In the last couple of decades, we've been reading genomes for the first time, actually understanding the information content in our body. Craig Venter sequenced the first human genome. I don't know whether you know this, but James Watson was that genome. And in fact, they found 3 fatal diseases in his genome, and he yet wasn't dead. Okay, so there was a lot more information in there than just those fatal diseases. We are seeing defects. We're seeing predispositions like BRCA1, for example, which predisposes a woman to breast cancer. We're identifying mutations that have happened in a cancer. We're detecting pathogens and why they're methicillin resistant, for example. And we're identifying susceptibilities. We're doing a lot of things with genetics right now that are very helpful, that we're now able to sort of open this book and read it. But we're moving on now. And what this session tonight is about writing genomes as opposed to just reading them. Writing the code of life is enormously powerful. It is a really big deal. I'm not sure you guys saw it, but Craig Venter recently created a synthetic organism that was completely new. He just reduced the genome to its bare minimum and created an organism. There are roughly 4 ways that I have read about to do it. Zinc finger, TALEN, CRISPR, meganucleases. There are a number of ways to do this, and there probably will be more as time goes on. But we're going to hear tonight from the panel about some of these techniques, what they're good at and what they're not good at, and things of that sort. And will it be accurate is one of the big issues right off the bat. This is a case right here where a CRISPR experiment was aiming, and let's see if I can do it here, at that green area right there. But it also hit all those red areas. So when we start checking out, grabbing a hold of a particular part of our genome, we have 3.1 billion base pairs in our genome, and there's going to be a lot of other potential matches unless that match is very carefully done. So one of the big issues that I see is, where are we going with this? Are we going to produce designer babies? Are we going to produce manufacturing workers that have 4 arms instead of 2 to make products better? Okay. We can have gene drives, things that basically take over a genome of a wild animal and basically can cause a whole species to go extinct at some point. The law of unforeseen consequences is the most important law we all live under right now. So, where are we going to be doing this? Where is this place going to have impact? In the human germline, where we are changing babies or fetal DNA, or even the very beginnings of sperm and ovums? Mutations during life, like cancer, for example, or aging or neurodegenerative diseases, of food-related products like plants, animals, insects, vectors, bacterial and viral organisms, microbiomes and pathogens of one sort or another, and then, as I mentioned, Craig Venter, synthetic organisms as well. So the purpose of tonight's panel is to talk about the power of this technology, and that's what we're going to do. So with that, I'm going to move on, and I'm going to actually ask Riley to come up, one of the first people in our group who's going to talk about his company, Desktop Genetics.
Thank you. I'm really happy to be here. I think this is a great opportunity to talk to a group of people who are possibly using CRISPR or other genome editing technologies in their work, as well as people who've read about them. It has been all over the media, and I think it's a great opportunity to sort of bring these two worlds together, which is sort of a theme I'm going to touch on a bit later. But just by way of an introduction, my name is Riley. Before Desktop Genetics, I used to work at Genentech as a bioprocess engineer, and before that, I worked in biological engineering at Dartmouth. And along with 2 of my classmates in graduate school, we started the company in 2012. We have 2 major revenue lines. The first is our DeskGen cloud software at deskgen.com. If you work in a lab, sign up today. It's free to use. Otherwise, if you want us to do the CRISPR for you, you can talk to us and our network of partners in our Advanced Genomic Services group. And ultimately, what we're trying to do here is, between our software and the larger network of suppliers and partners we work with, to be able to support you all the way through the genome editing process so that you can literally do desktop genetics. You can sit down at your laptop and create a custom cell or organism and be able to apply it to solve some problem. And we have 14 employees. We have over 2,600 users. We've done thousands of experiments, and we have quite a good success rate so far with those, which we're really proud of. I'm going to give you, by way of a background, the CRISPR talk. Just for those of you who aren't too familiar with it, I'm going to go through it pretty quickly, though, so I can get into some of the more interesting lessons that we've learned and some of the sort of stories from the trenches about being an entrepreneur in this space. CRISPR is an RNA-guided nuclease, effectively. You can think of it as find and replace, or for those of you who, like me, write code, something like sed or awk for the genome. And what you're going to be doing is taking an RNA component. It's got a variable region that you use to program this nuclease enzyme. That goes in and cuts the genome at a specific spot. This is the easy part. What's really interesting is what the cell does after. There are a myriad of DNA repair pathways that are then engaged. And depending on how this is done, you can get various types of outcomes. And those are actually what give you the editing event that you want. So CRISPR is precise cutting, but you still have to have the pasting functionality too. Why do you want to do this? You can go and you can fix genetic diseases, as we said. Recently, they published a paper where they went in and they were able to rip the HIV virus progenome out of CD4 positive cells in vivo, which is really, well, not in patients, in test tubes, which is miles away, as you all know. But it's still incredibly promising. We haven't seen anything quite like this, actually, in 40 years of biotech. They're also able to engineer pigs as factories for replacement organisms, and they're even able now to program the immune system to go in and target cancers more efficiently. So there's some really great things that people are doing here. It's also really, really hyped up right now. MIT Tech Review is calling this the biggest biotech discovery of the century. What you see is that the number of plasmids being requested, of publications, has had this massive hockey stick since about 2012 when this really broke. And we see thousands and thousands of experiments happening all the time. I think you'd have to read something like 60 papers a week now for the rest of your life to catch up and stay on top of the CRISPR research that's coming out. It's pretty remarkable. And so I kind of want to talk about why it's so popular a little bit. Well, one, you can do quite a lot with it. It's very versatile. It's got a lot of the same advantages of both RNAi and other genome editing techniques. But there's a little bit more to it, and we're going to come to that in a bit. But if you are working in the life sciences, it is a great tool. It does work, and you should definitely be checking it out. Our software also is up there. Briefly, rather than say too much about it, I want to focus on why is this necessary? Why do you need software in this space? And I think the best analogy I can give you is that if you've got something like a warp drive, you still need a navigation computer to go with it, right? Or you can, skip off a black hole or go through a sun. I think there's like a Han Solo quote along these lines. Well, it's the same in genome editing. You need a navigation computer for the genome to know where you're cutting and what the impact is going to be. And that's really why the computer and the machine learning algorithms behind it become quite important. So what have we learned so far? Well, first is that CRISPR does work with some caveats. What we found in our analytics is that human error is actually the greatest source of a failed experiment, and also that you really have to take into account the subsequent repair pathways that are being engaged. It's complex stuff, and we really do need computers to do it. The other thing, though, that people haven't touched on so much is CRISPR is popular in large part because it's very cheap to do. Academic scientists have to make every dollar count, and so when a company like Adgene is giving away the CRISPR vectors at cost, that's a large part of why you're seeing the 40,000 labs pick it up in a single year. And I think this is an important angle to keep in mind. One thing we also have learned in this space, now more as an entrepreneur, is that tools and services, of which CRISPR is actually quite a dirty word to a lot of investors, mostly because they don't think it scales. But I think there's a much bigger story here about the enormous amount of capital required. We've now seen 2 CRISPR therapeutic companies file for IPOs within 2 years, roughly, of being founded, which is incredibly quick by most standards. And so you have to think about, there's a sort of a much richer story here, I think, to discuss. The other thing that's still an open question is CRISPR a medicine, or and how personalized is it going to need to get? Is this actually more like gene surgery, where each patient actually needs a customized therapy? Because after all, everyone here has a quite unique genome. We don't know that yet, but if we do have to get that personal, I think we're going to really need to rethink regulatory, pricing, approval, distribution. It really challenges a lot of tenets of our healthcare model. And the other big thing here we see is that actually in bioinformatics, at least, data curation is actually the far bigger problem than anything else that we're seeing right now. It's a real mess out there. So if there are any bioinformaticians in the audience, I'm sure you can commiserate. And with that, other things they don't teach you in business school, recruiting is actually much harder than raising capital at the moment. Much harder by about a factor of 10. Every incubator skips this step, and they shouldn't. And there's this thing I call the cult of the pitch. Everyone likes to have a pitch session. They should really be having you pitch to prospective talent rather than investors, because that would actually get you a lot further. There's actually a major lack of cross-pollination between biotech and software. IT is generally an afterthought for most biotech companies. There are some exceptions, and they do some really incredible stuff. The SaaS model, also we've learned, is not a good fit for biotech. Biotech is an 18-month paper-based procurement cycle. SaaS model expects you to be able to load in your credit card details and be done with it. And these two things just don't fit together at all. We've lastly seen patent pooling. Everyone likes to ask about CRISPR IP. It's just one more instance of a much bigger problem here. Unlike other industries, biotech doesn't form patent pools. They don't cross-license as readily, though they have eventually gotten there once much blood has been spilt litigating. And I think that there's another big opportunity in this space as well. And then lastly, I think what we've learned is that the cost structure of biotech and the cost structure of a software company are completely different. Biotech is largely CapEx driven, whereas labor and brains is really the big cost that you'll have in software. And so with that, I think there's a lot of cool points to talk about, and I'd be happy to follow up with any of you. Thank you.
Just to take off where Riley, or start where Riley left off. So there are many applications for genome editing, and I'm going to try and move from the research space that Riley talked about to the therapeutic space. And so, but let's start with some of the principles that he talked about. Being able to target a specific gene and then make a modification in that gene is a permanent modification. And that's, I think, one of the key things as we start to talk about changing medical paradigms from chronic treatments to therapeutic cures. And I think that's one of the things I'm going to try and emphasize as we go through this talk. So I'm going to talk a little bit about Sangamo and what we've done. They asked me to give a sense of this, but then I'm going to jump into how and where can genome editing be used in disease settings where we hope to be able to move past even the most advanced of gene therapy efforts to permanent cures. So I'm going to skip over this. I think these slides will be available. The highlighted areas are really what I wanted to talk about, focusing on curative outcomes and then being able to do that through genome editing strategies. Ted's introductory talk talked about different modalities, different ways to target genes. We talked just about CRISPRs. I'm going to talk about an earlier platform, zinc finger nucleases. So this is a way in which every one of us, actually most every organism on the planet, has evolved to control gene expression. We have about 3 billion base pairs in us. Those break themselves down to about 30,000 genes. To go back to a tech metaphor, 2,000 of those genes actually encode the operating system of our genome, and those are zinc finger proteins. So a big percentage of our genome encodes zinc finger proteins. They're highly evolutionarily conserved, and each one binds 3 base pairs of DNA. What evolution did is mutate the small parts of these proteins in order to target or bind to 3 different base pairs of DNA, and then link these together in a way that they can bind to more and more complex or single sites within complex genomes. And that's what we've been able to do. Much like scientists brought antibodies into the laboratory and engineered those into monoclonal antibodies to target specific epitopes or specific protein sequences, we've engineered zinc fingers to target precisely the nucleic acid sequence we want. And then we can use that to bring to that site in the genome what we would call a functional domain. And now to bring that to the genome editing space, what we're able to do is bring to a specific site in the genome, driven by the ability to target or design zinc fingers that go to exactly the site we want, and provoke a double-stranded break. And that's really one of the key takeaway terms here, is a double-stranded break. We want to break the DNA at a specific site. And once we've done that, we can drive multiple solutions to that break, but the 2 principal ones are, one, we can cause that break to actually lose DNA and get stuck back together through a normal process that actually knocks out or stops the expression of that gene. And so that's a targeted knockout. We can also use that double-stranded break, because we know it's precisely in the genome where it occurs, to use a process, a natural process called homologous recombination, to insert large sequences or even small sequences, but sequences at that specific site. And this allows us to do targeted addition. But the key thing here is not just the outcomes that we can achieve, but these outcomes are permanent. They're permanent to that gene. And so if you do this inside a cell, if you do this inside the nucleus, the so-called endogenous genome in that cell, that modification is within that genome, and just like every other gene in that genome, it's carried through if those cells divide and into all of the future cells. So it's a permanent modification. And that allows us to begin to think about targeting diseases where we know that there is a disease associated with a gene. We can permanently change that sequence or disrupt that sequence. We can actually change the clinical outcome for that patient forever. 2 minutes? Wow, okay. So these are the kinds of things that we think about in terms of therapeutic outcomes. And then they asked me to talk a little bit about the company, and I've just a couple of slides. So how do you finance a company with these kinds of ambitions? There's dilutive and non-dilutive capital, grant funding, corporate partnerships that we've done, equity financings over the last 20 years, a combination of those. This is a quick sense of if you want to deliver or make these cures permanent on an in vivo basis, you use things like, was that ex vivo or in vivo? Oh, in vivo, okay. Yeah, well, these are somewhat backwards, sorry. So this is using this in the context of permanent modifications in stem cells versus existing strategies known as lentiviruses. We can also do this with AAV, which is another way of getting genes into cells, but those can be washed out, and the right-hand side of this is meant to tell you about the permanence of doing a genome editing piece versus a washout, and that's what's summarized here. And then back to what we're doing. So Sangamo is a company that's moving forward with the genome editing space. We've got 2 programs currently in clinical trials, 2 programs that have been approved by the FDA. We've got 4, actually 6 more programs that we intend to take through the FDA this year. So this is a technology platform that is reaching a sense of maturity, and happy to talk about this. These are just a couple of slides at the end to kind of compare and contrast various genome editing platforms, and I'm sure we'll have time to talk about these on the panel today. That's it.
We've learned a little bit about the technology here with these particular presentations. I'm kind of curious from each of you. Well, first of all, I'm going to give everyone a 60-second opportunity to introduce themselves. And Riley, you want to start with that?
Absolutely. So I'm Riley, CEO and technical lead at Desktop Genetics. Before this, I did as a bioprocess engineering at Genentech, so I made a lot of antibodies. Before that, I've always really been working in biotech. But I've also really had a strong interest in computing, sort of probably from playing too many video games. And so I've always operated, I guess, sort of at the intersection of these 2 spaces. And I think there's a lot both can learn from each other.
Great. Ed?
Edward Lanphier, the president and CEO and founder of Sangamo BioSciences about 20 years ago, and I've been in the industry for about 30 years.
Good.
I'm Eric Rhodes. I'm the Senior Vice President of R&D and the Chief Technology Officer, which has to do with genome editing at Horizon Discovery. I've had about 10 years in the genome editing space. Actually started at Sangamo and then moved through Sigma Aldrich, where we made available the first genome editing technology for tools, and now have moved to Horizon Discovery, which is a company focused on developing tools, cell lines, animals for use in research space, not clinical applications.
Okay. Jenny?
And I'm Jenny Rooke. I'm an early stage investor in life sciences technology companies. My training undergraduate was in physics and computer science, and then I found and fell in love with genetics. So I got my PhD in genetics, and then since then I've spent my career on the business side of biotech with a strong emphasis on genetic tools, such as these gene editing tools.
Okay. Thank you, Jenny. And I guess just to compliment everybody else, I'm also an investor in biotechnology and genetics, although I tend to focus on the diagnostic side. But what fascinates me about what we're going to talk about tonight is that if you've actually diagnosed a genetic defect, now you can actually fix it at the molecular level as opposed to giving it some kind of broad chemotherapy or radiation therapy to kill a cancer. So that's one of the things that fascinates me about what we're going to talk about tonight. So I'm kind of curious from each of you, what do you think are the most promising areas where CRISPR technology or TALEN or zinc technology is going to have an initial clinical effect? Not so much research, because this is mostly what we're talking about today has been research applications. But when is it going to actually affect human health?
I'll start with that because
Okay.
So we've been in the clinic with zinc finger nucleases for about 5 years in the HIV space. Very quickly, the product is an autologous cell therapy in HIV patients. So autologous meaning we take cells, blood cells, CD4 blood cells, out of a HIV patient. We genetically modify them with a zinc finger nuclease that targets a gene in those CD4 T cells called CCR5. CCR5 makes a receptor that's expressed on the surface of all of our T cells, in which HIV uses that to get into and infect T cells. Knocking out that T cell gene makes the receptor disappear from the surface of those cells. We then infuse those cells back into an HIV patient, and the goal is to create an active immune response to the virus, but cells that cannot be infected by the virus. And so we're in the midst of phase 2 clinical trials on that right now.
Okay. Any other thoughts?
I'll just say one other quick thing. So in addition to that, we also have a program in stem cells for HIV, and we have 2 open INDs now in the area of hemophilia and in lysosomal storage diseases. And then we expect to have INDs open for what are called hemoglobinopathies, which are beta thalassemia and sickle cell disease later this year.
Great, great. Eric, you have any thoughts?
I think one of the questions that we all need to ask ourselves is sort of what are the issues with taking each of the technologies forward in the clinic? And by far, Sangamo is the furthest ahead with the zinc finger nucleases, and they are really sort of setting the stage for these technologies that follow. There is only one technology so far that has been used clinically, talons, other than zinc fingers. And it has been successful, but there are still some of the same concerns about safety that there were with zinc fingers, and there will be with CRISPR and Cas9 as well. So I don't think to be honest, I don't think the game has changed with the advent of CRISPR-Cas9. I think that simply has opened the game up a bit wider to more players.
I agree.
I might also point to indirect benefits to human health. So unlike many investors, I do love the tool space for the impact that it can have on science and health. And some of the work that I funded when I was at the Gates Foundation was on genetic editing of animal models so that we could do better preclinical work in infectious disease, for example. So it wasn't using these tools to modify human cells, but modifying some of the tools that we need in order to get therapeutics into the clinic.
I'd absolutely second what Jenny said. I think for the vast majority of patients today, unless you have a quite horrific genetic disease or a couple other more tractable targets, the impact is really going to come from better medicines. There are a lot of diseases we don't actually know how they work. A lot of drugs fail in the clinic because we don't have the disease biology right, and the ability to more precisely figure out exactly how cancer is working or how Alzheimer's is working could change the life for billions of people.
Yeah. Actually, I'll add something to that myself. I'm an investor in a company called GeneWeave that was recently bought by Roche. And what they do is they genetically modify viruses that affect bacteria. And they modify them so that they light up when they find their target. They set off luciferase. And so they can tell when a person who's got Staph aureus has methicillin-resistant Staph aureus because they actually look for the MECA gene that's in there that conveys resistance. So they can now test every patient who comes into a hospital to see if they're carrying methicillin-resistant Staph aureus called MRSA, okay, which is one of the most serious clinical infection problems you find in hospitals. And it's just simply from genetically engineering, in this case, a bacteriophage, which is a virus that affects bacteria, so that it does our work instead of its own work. Anyway, all right, so I have a question here about how big is the market for clinical applications for gene editing, but I have the impression that it's pretty big now. In 30 years, it could be huge, and so I'm not sure how to answer that question from a time point of view, but I'm kind of curious, what diseases are sort of the most vulnerable? The single gene modification, those kinds of things first, or a pathogen that's got one variation that makes it drug resistant, or something like that? Any thoughts on that subject?
I'm happy to tell you where, everybody.
Yeah, you've already been talking about some of them, yes, with HIV and things.
So we've talked. The quote low-hanging fruit are genes where there is an unambiguous, and that's important, an unambiguous relationship between a mistake or that particular gene and a disease. So an example of that is sickle cell disease and beta globin. Another example is hemophilia, factor VIII. And there are many, many hundreds of so-called monogenic diseases that are directly amenable technically to genome editing strategies.
Right.
There's a number of issues, and you had some of these on one of your slides, that also have to be considered beyond just can I target a gene sequence and can I disrupt or correct that sequence. Much of it has to do with delivery and being able to deliver to a target tissue for the right period of time. Some of the issues that Eric alluded to in terms of specificity to those targets. So there's lots of different challenges, but basically the low-hanging fruit for the next 2, 3, 4, 5 years are going to be gene targets where there's an unambiguous relationship between the gene and the disease.
Yeah.
It's worth mentioning, though, in addition to, that's absolutely right, the vanguard, and certainly I think we all defer to Edward as the pioneer in this space, but I think an emerging category is also the manipulation of the human immune system to fight disease, whether it's cancer or infectious disease. And that is in the category that Edward's talking about of ex vivo, where you, at least to start, where you remove components of the patient's immune system, modify them, retrain, for example, components of the immune system to go back in and fight cancer cells. I think that's a very exciting area as well.
Yeah, I think that's true. In effect, what it comes down to is we have been living for a few billion years here with evolution kind of controlling this manipulation, and now all of a sudden we're grabbing the steering wheel, which is kind of a scary thought, and we'll probably start off at first by just simple things. We don't want to go too complicated right off the bat because of the law and unforeseen consequences that I mentioned earlier, which means all of a sudden we could extinct an entire species of mosquitoes or something like that because of one simple gene drive or something. Okay, I've been reading a lot about the IP battles that are in this space because it sounds like CRISPR, for example, there are two different organizations that feel they kind of invented CRISPR and both basically have pending patents of one sort or another. Do you guys encounter in your companies or in your investments, do you encounter these IP battles and how do you basically deal with them?
We do, but it's usually, it depends on the customer and whether or not they have, some companies just have enough money to go out and license all of the IP and be done with it, which is a great strategy if you can afford it. You get actually quite a huge range. On the other end of the spectrum, you have the academic researchers who don't really care one way or the other, and so they'll go out and do CRISPR because they read about it and they'll try it out. In terms of us, we're sort of a bit removed from it, and so the larger effect is whether or not they're ready to move with CRISPR. They sort of have sorted their own stuff out internally and they feel comfortable with it. But some of the companies have really impressive programs, whereas others are sort of getting more comfortable with it.
Any other thoughts? Yeah.
Horizon is pretty much right in the crosshairs of the IP situation in that we provide tools for research now. I think we see a different outlook from those groups that are on the therapeutic side. Clearly, there are 3 major groups in the CRISPR-Cas9. Sangamo really has the zinc finger tied up. There appears to be mostly one group about talons. But in the research side, where people want to use all of these, and we employ all of those technologies, we've had to take licenses to all of these things. And it is complicated trying to understand what will rule and what are our customers demanding of us. So we've had to be as broad as possible, and we've taken licenses to virtually all of these technologies.
So Jenny, I suspect this is one of the things that adds risk to every investment you make.
Right. Well, I'm an investor in Caribou BioSciences, which is one of the leaders in the CRISPR space. They have the exclusive license to the UC Berkeley Jennifer Doudna IP that if you've read about the space, you've probably heard about. So there's certainly, we spend a lot of time thinking about intellectual property, as many life sciences, innovative life sciences companies do. More time talking to lawyers about intellectual property than average, I would say. But what I'm seeing in the space, not speaking for Caribou, but just as an investor, is that there's so much excitement about the power of the technology and so much interest in starting to use it to make solutions that companies are taking, I think, the stance that in the long run they'll figure out a business solution and let's go ahead and pick a problem they want to solve and try to use whatever the correct tool for it, and then hopefully retroactively we'll figure out some appropriate business relationships to enable those. So yes, it is complicated. It will be years before that particular dispute around CRISPR is resolved, because that's how long it takes. But in the meantime, it's delightful to see that it's, as you're saying, Riley, definitely not holding back research and also not holding up progress, I think, in the commercial sphere as well.
I also noticed that there seem to be different regulations in different countries as well, so that all of a sudden somebody in England may be able to do something, or somebody in France or China or something can do something that we could not do here at some point. So that's another challenge, is that some countries may be a lot more liberal about this and some countries may be a lot more paranoid about it.
That's true for both the intellectual property and, I think, as you're saying, for the regulation of the use of the technology.
Yes.
So, for example, how the different countries think about whether using CRISPR-Cas for gene editing counts as genetically modified or not is different.
Yeah. And what they're doing it on. If they're doing it on a fetus, on a human germline, that's a whole separate issue than when they're doing it on a piece of corn, corn or something. So, actually, moving to the FDA. Have any of you had experience with how the FDA likes to look at this? Because I would think the FDA, since they've been so incredibly difficult about devices and about drugs over the years, that they would be horrified by this. Because they're the people who are supposed to be the risk managers, the ones that eliminate all risk before the public population can use it. So have you encountered FDA resistance to this?
Everything we do in terms of therapeutic product development is regulated by the FDA, and I might be the single anomaly on the planet, but I think the FDA does a spectacularly good job.
Good.
They have been more partners than regulators. They are curious, they're engaged, they're thoughtful, they're rational, they're reasonable. I think they're wonderful.
You may be the only person I know who thinks that.
And that you and the FDA that are sitting in the back. No, they really, they have FDA has a terrible reputation, in my opinion, in the area. What?
They have a tough job.
They have a tough job, but
yeah, they
have a bureau that's CBER, the Center for Biologics Evaluation Research. They have a Center for Gene and Cell Therapy, and within that, all of the genome editing work that we do and others do is housed. And it's a scientific regulation agency. They could use more funding. They have turnover. There's lots of issues related to a bureaucracy. But and they balance risk-benefit. They ask those questions. But they're a thoughtful and engaged group that, I'm pleased to have I'm pleased as a citizen, I'm pleased as a consumer of medicines, I'm pleased as a parent that children that get medicines to have the FDA do what they do.
Yeah. Other experience, Riley?
So I think that the FDA is an excellent partner if you engage with them and engage with them early, especially if you're a small biotech business. But I think they are a perfect example of this almost immune system the biotech industry has against people coming in from tech or hardware or devices who don't know this, getting in some really big fights. And I think Theranos and 23andMe are up there at the top of the list. They effectively flout the FDA's authority, and they do not like this. And they will come after you very hard.
Yeah.
And so you have to be very careful. And people don't, they're like, oh, regulatory, we're agile, we're going to innovate. FDA is not like agile. They insist on waterfall.
Yeah.
You cannot, they don't like minimum viable products. It has to be version 1.0, and that's it. And this is a complete cultural shift, though, for other industries.
Yeah, I think that's true. It's interesting that they have approved drugs in the past and then later on withdrawn them, like Vioxx, for example. And the fact was that Vioxx helped 100,000 patients but hurt 5,000 or something like that. So they often are concerned about removing all damage before it's approved, even though it might benefit 95,000 of those people.
But this is a direct side effect of what happens. If the FDA does their job they don't get a bonus, they don't get a reward, they don't get stock options or an IPO. But if they do their job wrong, they get dragged in front of Congress and raked over the coals.
Sure.
So what do you get?
And if you do your job very well at the FDA, people will start going on cruise ships and getting their procedures done there, or going off to Malaysia or something like that. So they have to be a little careful that they don't drive all of medicine overseas. When you're told you have colon cancer and you're going to die, you're probably going to find a solution even if it isn't FDA approved. The accuracy associated with these technologies, I'm not sure that we fully grasp what the negative byproducts, the negative effects can be with a lot of these things. Yes, you can fix a monogenic mutation, but if you also disrupt something else elsewhere in the genome, what's the side effect you're not going to notice? Like they're suddenly much more prone to colon cancer 5 years later or something like that. Any comments on those sorts of things about the accuracy of these?
I wanted to emphasize something that Riley, I think, very correctly he made in his talk, which is this is a tool whose accuracy is wildly different in different contexts, including, in particular, the hands of the practitioner. So if we were talking about scalpels have just been invented, you wouldn't talk about their lack of accuracy because I can't do open heart surgery. Right? I'm not trained, I don't have the equipment, et cetera. The same is true with CRISPR-Cas. It's really easy to pick up in the same way that a scalpel is, unlike some of the previous technologies which had a higher barrier to entry, the level of expertise necessary to even give it a shot. So we're seeing a much wider variability in accuracy and an impact there that is one of the biggest knobs is the practitioner and in the cellular context. So some of the work that Caribou is doing internally on the technology development front is to really understand the relationship between, for example, the guide sequence that's used to target the place where you want to make the break in the genome, and how that varies depending on what type of cell you're in or how long the guide sequence is. And it turns out all of those variables matter quite a lot. Not everyone knows that, right? So you're going to get a wide variety of results. So you shouldn't take either the range that's visible to the public or any given example as representative of the accuracy of the technique.
And I think you have to test the technique on many people, not just one or a few, because the fact is everybody's somewhat genetically unique, and you may find a side effect in a person that you didn't expect. Okay, so it is a complex issue. Yeah.
Absolutely true. The other thing, though, I would add to that is that the current generation of off-target detecting assays push the limits of next-gen sequencing systems to the borderline of their capability, except that they actually don't always agree with each other, and it depends on who actually ran the sequencing assay that day. So there's enormous variation happening in here, too. So now what does a false positive actually mean? Yeah. Is it noise or not? And then you touch on an extremely, extremely important point. Everyone has a different genome, and there are now many cases emerging in the data of examples where a different genotype creates a very attractive off-target site that is picked up in one context but would not happen. Right. And in a clinical setting, this could actually kill someone. We're talking about a situation where if you don't genotype the patient, you could actually cause quite a bit of harm to them. But unfortunately, our system doesn't even really have a constant for wait. We have to rediscover the drug for every patient. How does that work? It's going to be quite a challenge.
I'm thinking about Vioxx again, and the exact same thing, that it worked really well for a lot of people, but there were a few people it didn't work so well for. It actually caused more problems than good. That actually is interesting, because what it comes down to is that we're going through this gene editing revolution at the same time as we're coming to get our arms around the personalized aspect of medicine, that everybody's a little different at a genetic big data level. And so that we're going through both these things at the same time, I think that's one of the big challenges that we have, is that we're having companion diagnostics that make you take this drug, but someone else takes a different drug for the exact same condition.
I think that's also why you're going to see this field develop with ex vivo therapies to start with.
Yes.
Right.
You pull the cells out, you do your manipulations. There are at least a battery of checks that you can do before you put them back into the patients. Yeah. Once you start injecting directly into the patient, you need to be pretty sure of what you're doing. Yeah. And there's a different safety protocol associated with that. So I think the steps are happening in that order for the most part. And depending on the seriousness of the disease or the consequences of the therapy, we will move certain things into an in vivo setting before the ex vivo.
Yes.
And that's where, and Eric's right, that's where we've been, is in an ex vivo setting, but now we have developed the kind of specificity that you're talking about and the ability to characterize both the on and off target activities to the point where we've been able to move to an in vivo setting, so directly into the patient and in a way. And so those of you who are interested in that kind of question of the kind of specificity, the kind of rigor that one has to do to show that you're targeting precisely the site that you're interested in and doing nothing else.
Right.
The National Institutes of Health has a committee called the Recombinant Advisory Committee, the RAC, and novel genetic therapies go before the RAC. And so we've presented all of our ex vivo, but now most recently all of our in vivo genome editing work at the RAC. That's up on the NIH website, so it's all there. And much more data in terms of specificity, off-target activity, toxicology, carcinogenicity, et cetera, go into the filing of an investigational new drug application with the FDA. And we've now have 2 approved in vivo INDs that we're moving forward. So it's an important question, but if you're capable of building highly, even singularly specific reagents, it's a solvable issue.
Yeah. I think what we may see is a lot of work in animals and plants first, okay, and working on things that we can modify, see how well it works, see if 1 in 100 cows die if we modify the cows to make low-fat milk instead of high-fat milk or something crazy like that. I mean, the fact is that we may start off by not actually functioning directly on humans.
But Ted, to a certain extent, that's already been done.
I know it has, yeah.
And Eric was at Sigma. You guys made hundreds, if not thousands, of transgenic animals with different models. I think dozens, it's probably now hundreds of plant species have been modified through genome editing. So if that's the prerequisite, those prerequisites are finished.
Yeah.
And we are now moving to the point where this is being used and will be used both in cell-based and autologous cell settings, but also in vivo genome editing.
Yeah. There was an interesting comment that someone made here that gave me as a suggestion. The stem cell revolution happened about 10 or 12 years ago. It is not actually sort of swept through like everybody thought it was going to sweep through. We're still not actually making hearts or making livers, even though we knew roughly how to do that sort of thing. Craig Venter sort of sequenced the genome 16 years ago or so, and yet we're still trying to figure things out. How long do we think this is going to happen, that we're going to play with this and mess with this and experiment on animals and experiment on severe cases or monogenic diseases and things like that before it actually starts becoming a commercially big deal as opposed to a interesting piece of science?
I think that's not a scientific question. I think, that's a business question. It's a regulatory question. I think that framework is really what needs to happen. But I would say we've been working on gene therapy for 40 years as well. We've killed some people along the way, unfortunately. We got it really wrong in some cases. We've learned a lot of lessons, at least. And now there's a lot more safeguards in place. And I think it does, but I would say does it end? What is done even look like here, right? Is it any gene, any time, every time? Complex multigenic things like Alzheimer's and cancer? That might be decades away, but that doesn't mean you can't have an impact here and now.
Any other thoughts? Yeah.
Well, I think it's there are active clinical trials, both with platform we're working on. There are clinical trials with CRISPRs that have been announced for 2017, maybe even earlier. There's been at least one patient treated with a tau-modified product. We're now at the point where it isn't a question of, gee, will genome editing be something? The question now is, will that therapeutic product show safety and efficacy in well-controlled clinical trials, just like any other drug?
Yeah.
So I can't tell you that that drug is going to work, but the targets we're going after, if we know that replacing factor VIII in a hemophiliac will improve and normalize clotting, then if you can do that inside the patient, there's every reason to believe that that can be effective. So there are scientific platforms But once those platforms have been translated into a a drug that can be reproduced on a lot-to-lot basis, just like you do in GMP manufacturing, it now becomes a function of, can that product stand up to a statistically significant, well-controlled clinical trial that shows safety and efficacy, just like any other drug.
Mm-hmm. This is such a powerful technology that I worry if you get some psychopath using it, that it could actually be weaponized in some fashion or another. Has there been any thought to that? I mean, right now we're basically depending on the fact that everybody working with CRISPR and Talon and zinc finger is well-intentioned.
I worry about that too, but even as a weapon, depending on the sophistication of the person developing this thing, you've still got the issues of delivery, specificity, all of these kinds of things that plague the therapeutic space, or at least are the challenges in the therapeutic space. So, these things will evolve together, I think, but we have to be vigilant and careful about what we do.
Yeah, we have to be ready,
I
think, to a certain extent.
A lot of potential to do bad things if you're not careful.
Yeah.
You mentioned gene drives in your introduction, and which is not really part of the sort of genome editing as we're talking about here. Is a bit tangential. But I think it does highlight something very important, which is that these are very slow-moving things. Mm-hmm. Gene drives happen over generations, so they are the century-long weapon. Mm-hmm. Same thing even in this case.
Gene drives in large mammals. Gene drives in insects can happen a little quicker.
Fair point. It's based on the generation time. But my point being that even genome editing, if it were a weapon, this is not a bullet. This is something that's going to take place over weeks or months potentially. And so they're not actually terribly even effective in this case. Mm-hmm. And I think that's something that's just important to bear in mind. The kinetics just really aren't there.
Okay.
Nature's already a pretty good mutating mechanism.
Yes.
Mm-hmm.
So
cosmic rays do a good job, yes. Do we have to worry about the high school chemistry student going off and deciding he wants to do something crazy? In other words, if CRISPR is so cheap and so accessible and you can get things done, is it possible that you can have some rogue high school student going off and doing something? Yeah.
I can say, having worked with almost 100 different companies, the level of support that even skilled people need at times is surprising. Right. So I'm not horribly worried about, maybe your honor high school student is something to worry about, but your sort of average, mean, mad at the world sort of high school student, I'm not sure it's exactly there.
But I mean, if one really wanted to do something biological, mm-hmm. there are far simpler ways to use viruses that don't require CRISPRs or genome editing or anything else. So
the synthetic biology space is facing the same question. They can
hundreds
just
of ways to
design it on paper and put it into a machine.
Right.
So.
Right. Okay. Sort of as a last question, is there anything that the U.S. Congress or the U.S. government needs to do to put sort of rules in place to prevent people from doing certain things like germline modifications in designer babies, but on the other hand, to promote the idea of healing hemophilia or something like that? I mean, I'm kind of curious because right now I don't think Congress understands this at all, okay? And so I'm wondering, do we need to put in some rules, some limits, some boundaries?
So I think the framework is already in place, right? This is a new version of gene editing. This is not gene editing has suddenly existed where it didn't before. Again, Sangamo's been doing this for decades. We've got other kinds of gene editing tools. So this is already contemplated by the RAC, the FDA. I don't think it changes the framework.
Okay.
I'll do this part quickly, but in addition to my day job, I'm also the chairman of an organization in Washington, D.C. called the Alliance for Regenerative Medicine, which is an organization with about 300 members, all of the gene therapy companies, all the genome editing companies, all the stem cell companies, all the cancer CAR T companies. So it's a large organization, patient advocacy group, so on. And we were early on, a little more than a year ago, calling for a summit on genome editing in fertilized human embryos. Congress has gotten interested in that. The National Academy of Sciences has gotten interested in that. More recently, this last, what month are we in? March, April. Thank you. This last winter, the National Academy of Medicine, along with the Royal Society of London, or of England, and the National Academy of Sciences of China did a summit in Washington, D.C. on the whole issue of genome editing and to the germline. So there is active political discourse around issues related to gene therapy and genome editing. You're right, it takes a lot of education in Washington to do that, and an awful lot of time sometimes.
Yeah.
But it's important that one does have make an effort to try and educate politicians on these issues.
Okay, okay. All right, I have some questions that basically have come from the audience here, so I'm going to actually throw them out. I haven't filtered them in any way, but I'm going to sort of go through them. First one actually is kind of interesting. It says, the panel seems to be in agreement on just about everything. What is something that you are all in grave disagreement about?
Well, we're all in subtle disagreement about which one of the platforms is the better platform.
CRISPR, Talon,
Yeah, or
Zinc
this or
Finger,
that,
yeah.
or this or that.
I doubt it.
Yeah,
Right?
well,
So
no,
I disagree that we disagree, because I think
I completely disagree. But I think if you scratch the surface hard, you'd find, camps relative to where one thing is better than the other and that sort of thing.
Yeah, that's what
I
I'm
think there are
curious.
pros and cons to each. I have funded every single gene editing platform we're talking about one way or another. CRISPR is just the most recent, and so I think each has incredible strengths and drawbacks, and we will, in 50 years, have a collection of all of them doing different things better and worse.
We use all three.
Yeah.
And we're agnostic as to which one we use. We say, what's the application? What are we trying to achieve? Which one serves best in this particular situation?
We could use all three, or rather implement them. We don't, because no one asked for them. They asked for CRISPR.
Right.
The research scientists, which are the vast majority of the market.
Because it's best for research.
And it's also because of the economics, though.
That's why it's best for research.
Point is yeah.
And the technology is not going to stand still. There's going to be a CRISPR at some point.
Yeah. We've already had new nucleases,
Yes,
for example.
it's going to happen. It's going to happen. We're going to move on. We're basically just getting our hands on the accelerator pedals and the brakes associated with modifying genomes. And there are going to be new ways of doing it, no doubt. So, so when would we be able to design totally new mammals for pets? And
You guys did.
I would make the argument that we've already done so, because I don't think a tiny miniature poodle is actually a natural animal. Okay.
Sigma did something.
So yeah, there's a glow cat. That
A
has
glow
been
cat.
made.
Yes, I've heard about that one. Yes.
And there's TV shows about glow everything animals now.
Yes.
There's glow fish commercially
Yeah.
available too.
Luciferase is
Didn't
an
you guys
easy
do
one.
some, the
We tried,
hypoallergenic
yeah,
stuff?
we've been working, or when I was at Sigma, there was work being done on a hypoallergenic cat by eliminating the dander gene. I don't know what's happened with that, to be honest.
I don't
So,
either.
I mean, those are some of the simple things. Again, there's more of a medical push than a commercial push. The glow fish, obviously, was more of a commercial novelty. I think that the concept of the hypoallergenic animal is something that would have some benefit, but it does take some investment, and there's regulations even in the domestic animal field. So, I don't think there's quite the commercial push to do
Our
that.
old-fashioned way of modifying genomes by sort of selectively breeding has formed a whole bunch of dog breeds, for example, that dislocate their hips all the time.
Yeah.
So,
Yeah. That raises actually a much more interesting question, though, which is that biology actually isn't very easy to design. It's very easy to evolve, right? Anytime biology actually works, it's usually in the context of some sort of evolution or synthetic evolution
with
process.
some time involved.
Yes, over multiple generations and iterations, because effectively it's a simultaneous optimization problem. We're actually very, very bad at designing biology from the ground up.
Yeah. But maybe in 50 years we'll be much better at it.
Well,
Although-
so one of the companies I invested in is called Zymogen, and part of the, I don't think they would describe it this way, but part of why I did so is because their approach, which is very high throughput, very broad number of tries at once, so a shots on goal strategy, admits that we're bad at designing things, right? And so it says, let's try a bunch of things. That's straightforward if what you're working on is bacteria that can produce chemicals. It's a little harder if what you're trying to make is 10,000 cats, and then you're going to select the one that meows the way you want it to. Right? That's a different optimization problem. So whether we can automate and do shots on goal for pets, I think is a kind of a different infrastructure investment. But I agree, it's not the design problem, it's a selection problem.
Well, and I could make the argument too that the microbiome we all carry around in our guts is also a pet. Or many, many pets.
It might say that about you, actually.
Yes, exactly. In fact, I think they're going to say I'm basically the house, and they're the occupants of the house.
You're the meat car.
Yes,
right, yeah.
We're all villages, effectively. Yeah. And so there's a lot of risk in that situation too. The microbiome is we don't realize the extent to which we cannot live without a microbiome to a certain extent. So, okay, here's a question that I think I somewhat touched on before, but I'm going to ask it again because this is a more specific one. What is the opportunity for a kid using a $70 CRISPR kit disrupting the biopharma industry? Or should these kids go after making genetically engineered biofuels?
So one thing I can say, starting an early stage company in this space, is that the barrier here, the $70 kit gets you barely one step. It's actually the $400 million pilot plant you would need to go into production. There is no Amazon Web Services of biotech really yet. In software world, you can get servers on demand, capacity on demand. It's trivial now. Right now, you have to build your own. It's as if every tech company had to build an entire data center on the scale of Google to even sell one instance of their product. That's the challenge right now that you're facing. And so that's far and away the much bigger barrier.
Well, I do know that there are a lot of cloud-based genetic tools at the present time where you can buy just a few minutes on DNA Nexus or something like that and do an awful lot of work.
For compute, yes. And there actually are some really exciting companies like Transcriptic and Emerald with their cloud labs that are also really cool.
Right.
But this is new stuff, and I think they will definitely change the arithmetic here.
Yeah. This is an interesting question because I know Craig Venter's been focused on this one. Does CRISPR promise a dramatic life duration extension? Human longevity is a very interesting issue. We are all on this earth for a certain amount of time, and we tend to degenerate as we go. But is there possible that I could someday be genetically engineered to be a teenager again?
So you got to remember, what we're talking about here are tools that can be used to manipulate biology. So now, ask the question differently. Is there biology that one can use to extend life or to create a next outcome? If there is, then these tools might be useful to insert or delete or manipulate. But de novo, these tools aren't going to do those kinds of things.
Yeah. And I think aging is quite multigenic. It's not
well, I mean, again, multigenic or not, that's the biology of the question.
Yeah.
If there's a genetic basis for that biology, then these tools can be used to affect that outcome.
I think these tools are helping to drive the research down that path and doing a much better job than what we had 10 years ago. But I agree with Edward. It's biology that lies at the heart of all of this.
Yeah.
And every disease you tackle, oncology especially, where we look a lot, every change you make has some other effect, and the cell is very adaptable to those things. And so we have so much we don't know about so many diseases. The monogenic diseases we can associate with a disease, but it doesn't mean that if we fix it, that patient will necessarily be well. They may have already adapted in another way that gives us some sort of negative outcome. So who knows? I think it is very difficult.
And with immunotherapy and things, we're trying to get our hands around cancer. So the next one that we probably have to focus on is neurodegenerative diseases, where we are just simply aging poorly and turning into zombies, effectively. Yeah. So it's an interesting place we are, because in effect, what we are doing right now is taking over Darwin's evolution with our own personal design. For millions of years, effectively, life has basically gotten smarter or gotten better or gotten more competitive or fed itself by using evolution, using survival of the fittest, effectively. Now all of a sudden it's survival of the smartest or the survival of the best tools, effectively. So Do you see a role for machine learning, for artificial intelligence in this? And I think the answer obviously is going to be yes, because we are dealing with such large data sets that we're going to need help.
Yes, that's what we do. That's like our company. So yes.
Yeah. Any other thoughts on AI? And actually, I think we better hurry up and do these genetic modifications quickly, because AI is going to take over pretty soon, if I believe Elon Musk. But that's a whole separate issue.
I mean, it's being used. We're working with Desktop to try and improve the technology itself. And then obviously the whole question of biology and all the impact of all those big data sets. So it's a yes.
Yeah, I think I'm more on the Ray Kurzweil side of saying that AI is going to be helpful to us in figuring all this out eventually, as opposed to the anxiety-producing approach.
I think machine learning and the larger suite of tools to handle big data problems and opportunities is a great way to identify new questions and hypotheses out of biological data, and then you still have to do the science.
Yeah.
So.
Yeah. The next question I've gotten from the audience is, I'm a junior in high school. How can we have this taught in our school?
See me after. I'm happy to do that.
Sangamo still have programs, right? For high school genome editing. There was at least a program at one point that was being put out. So I think they're adopting it. And when we go to these national conventions and things like that, there's generally an education sector where you have high school level, in some case junior high, learning about these seminars about the technologies, what they can do to obtain that. So I think it's happening, not everywhere. Get your teachers involved.
Yeah. It's 5 or 10 years behind what's going on in the laboratory of the most advanced companies, I suspect.
Oh,
yeah.
Certainly.
Yeah.
And it takes a while, and there is equipment needed. I mean, as Riley said, you can buy the $79 kit. You still need to be able to grow cells, whether they're bacteria or mammalian cells. That requires other equipment, and it gets more and more sophisticated.
Well, this next question is actually asking about why is it that CRISPR is so special? Because zinc finger and talon and other things, nucleases have been there before. But CRISPR, I think the reason is, and I'm going to be interested in what your response is that it's cheaper and easier to do.
It's cheaper and easier, and in particular, I've used before the analogy of it being like the invention of GPS. There's now, with a long enough sequence, an unambiguous relationship between your directions and where in the genome you want to hit. Whereas before we were reduced to saying, when we wanted directions to someplace in the genome, you had to translate through protein, which if you've got the skills is an incredible way to do it because it's very specific and very amenable to therapeutics. But if you don't, and maybe you're not from the area, you're reduced to saying, well, I think you go down to the Dunkin' Donuts and you take a left, but then there's 2 Dunkin' Donuts, right? So there's room for lots of error if you have to go through the process of translating where you want to be in the DNA via a protein-based direction system. So CRISPR is really exciting because there's a one-to-one relationship between the guide sequence DNA and the target sequence, and so you're not trying to figure out how those things relate to each other, and that's part of why it's so straightforward to use for new practitioners, because they don't have to go through the protein step.
Mm-hmm. Next question is, how do we get involved and make money if we are interested in riding the CRISPR wave?
Caribou is hiring pretty aggressively, so is Zymogen. So I've got some great portfolio companies who are looking for talent.
I think this is actually an interesting point, though, because CRISPR is so cheap. You have to think of the business models a bit differently. And I think they're coming at it more from the informatics side. There are 2 real issues. The first is that the margin on the CRISPR reagents is being put under tremendous downward pressure at the moment. And so it's very difficult to say, like, oh, well, we're going to get a commission and we'll use the software, say, as a loss leader or something like that. The other issue, though, is I think there is a lot of resistance, especially in academic laboratories, to paying for software informatics because, after all, I have a postdoc come in and do this, and I'll have someone from that bioinformatics department come over and do it. And we were going to save our money for tangible goods. There's quite a lot of bias there, if I can be so blunt. And I think that does make it a bit more difficult. I do think, though, that there is certainly a lot of opportunities, but you have to just be a bit more creative about how you seek them out.
Yeah. So the so what I think from that, and that's absolutely right, is join a products company, a company that is focused on solving a particular problem that is associated with a market and is using CRISPR as part of one of the tools. If you are interested in therapeutics broadly, there are options, as we've heard, outside of CRISPR that are more broadly about using these tools for human therapeutics. If you're interested in industrial bio or in ag or in research and you're agnostic, then you might look for the leaders or the emerging leaders in each of those sectors that are using CRISPR as part of their overall strategy.
I think if you understand the technology as well, you just look for an unmet need and decide whether or not the technology can address that unmet need. And I think that's been done to a degree because of the other technologies that are out there. CRISPR is cheap enough now it's a lower barrier, at least, to entry to try some of these things. So I think, if you have a great idea, you can get it funded, but it's that great idea coming up with it. What is the unmet need that you can address with this technology?
Sure. Okay. I'm looking at the next question here. Changing the genome of cells in an adult organism is a tougher problem than changing isolated cells. Could you please comment on the state of the art in mass genomic conversion? And that is one of the challenges. Ed, you want to try that one?
In vivo therapy.
Yeah, that's...
Well, it is. It's more complicated. It requires many other components beyond just the genome editing piece of it. It requires novel or perfected delivery systems. It requires tissue-specific promoters. It requires the ability to affect the duration of expression. So there are many, many, many other levers and many other elements that go into being able to use genome editing systemically or in vivo versus isolating cells, modifying those cells, as Eric said, being able to characterize those cells before you put them back in. So
most of the gene editing therapies, whether or not they're, well, I should say gene therapy applications, are generally still very targeted. You're not trying to affect the whole body. You're looking at trying to get an effect in the liver or maybe isolated to brain or within the eye.
right.
Yeah.
So I don't think anybody's really thinking about whole body, modification delivery is just an almost insurmountable challenge. But
It does suggest, for example, that we can harness viruses to do our work instead of their work.
we already do.
We already do.
And there's some
Most of
great
gene therapy.
Even in oncology, I understand as well.
There's some great meta work where researchers are using CRISPR to, in a directed evolution work, modify viruses to have different tissue specificity so that they could then deliver
Target
gene
cell
therapy.
tissue.
Yeah, it's pretty great.
Yeah. There's an interesting question here. Since DNA is data, easily created, copied, do you see the IP landscape changing in the future, the way IP is handled more similar to the software and digital world? And one of the things that's interesting about this is that, Myriad thought it had a patent on a particular gene sequence, and then the courts decided all of a sudden you can't patent a naturally occurring sequence. You can only patent an algorithm that puts a lot of them together or something like that. So that is one of the interesting issues here. Any comments on how the IP landscape is changing in this area?
I think as biology becomes more of an engineering discipline, this will need to change. I think that the way we've approached it so far isn't quite the right fit. And I think that there's a level of sophistication that we're going to get to in the near future around this topic.
This is maybe an area where we have room for disagreement, which is that, so as someone who came from the coding world, and I was studying computer science, and then I got into the lab my first year in graduate school, and I tried to do PCR, and it didn't work, and I was incensed. What do you mean it doesn't work? I designed it correctly, I followed the protocol. It turns out DNA is code until you get in the lab, and then it's DNA. Right? It's a molecule, it has properties, we don't understand it, it works in a very messy context of human or other biology. So none of the products that are going through the clinic are data, they are physical things that have to work in these messy environments. So that's, I think that it's interesting to rethink how we deal with IP there But I don't think it's as simple as that it's just information. I think the IP that stands is around products that people have done the work to have biological insight, take them through the clinic and show that they have human health benefits, or show that they make improvements in how we do agriculture. Right? That's where the invention is. That's where the business is.
When will we target germlines for improvements, and will there be a split in our focus, our species, on those who have been edited and those who have not?
Well, people already have tried with CRISPR,
Yes.
right?
Yes.
I think that is important to point out. And the problem that happens is that you get a mosaic embryo, effectively like a Dalmatian embryo.
Yep.
And so that it has to do with, even at the single cell level, by the time it divides out a couple days later, you're getting different outcomes in each of the different cells, and so you're not getting a complete conversion. And this is why it was more or less unanimously decided it's a very bad idea to do this. It's just not safe, it's not wise, and it's not really in line with the current scientific practice.
But I think that's a matter of time. Right?
Yeah, I do
That's
too.
a current technology limit, and we could guess about the timing on that. But we are currently advancing the tools for other applications that will make it absolutely possible to do germline modification, either in vivo whole body transformation, as someone already asked, or ex vivo modification of single cells or blastocysts or embryos. So that's going to be possible. So that's the question, I think, stands, what happens when that's possible, right?
Yeah, and I agree with Jenny. I think the findings out of the summit were go ahead and do research in embryos, human embryos, but don't do any implantation. So the techniques, as Jenny just said, will be perfected tomorrow, a year, 10 years, some time unit. It will be perfected. There are, cultural norms, there are legislative norms, there are funding agencies, there are all kinds of ways to try and regulate the use of genome editing in fertilized human embryos, but once it's perfected, it will be used. And the question is how and what gene targets. Goes back to the biology, right? We're not going to create biology, but we can direct and modify biology in a permanent way.
Yeah.
And as much as I may not like it, and I may be dead by then, but there will be genome editing in human beings.
Actually, I can tell you one of my companies, Natera, does this already. In vitro fertilization, they typically fertilize multiple embryos, and then they selectively reimplant some. And they could reimplant only the blonde ones, or only the tall ones, or something like that. Right now, they only implant the ones that don't have Down syndrome, or something like that. But they could very easily be doing designer babies with just
Pre-genetic
IVF.
diagnostics is a
PGD.
is a quote acceptable way to go about with families who have Huntington's or other diseases.
But they can be
It's
selected.
a very reasonable way to go about that. It's a whole different issue to go in and then say we're going to target a gene to provoke some kind of biological outcome.
Right.
But I do think it will happen.
We're starting to run out of time, so I'm going to go very quickly through the last couple of questions here. One person has asked, can we cure mosquitoes from carrying the Zika virus, for example? And I believe that is already being done.
Discussed.
And being done for malaria as well, I understand. So I think the answer is very obviously so. And in that case, you're modifying a mosquito's gut. You're not modifying a human's gut. So it's a relatively safe one.
I think the issue there is, can it be done? Yes. Will it be done?
Yeah.
And how broadly will it be done is a political question.
Okay, I think we're close enough toward the end of time here that I'm going to ask everybody to kind of give me a sort of a wrap-up comment on this thing very quickly. Jenny, why don't you start if you want? Just to, where do you think this is going? Okay.
I think the one thing that we know is that we don't know. And that the part of why I'm so excited about CRISPR based gene editing tools is that it has captured the attention of many people who have never thought about genes at all, right? And that's both in the public as well as other types of engineers and scientists and researchers. And I think that bodes incredibly well for science. It means the set of people who can come together to think about the hard problems that are facing us for the next 50, 100 million years is much broader, is much more interdisciplinary, and that I can't possibly predict what it means to, as you said, Ted, suddenly have a much broader and stronger grasp on the tools of changing our genetics and also all of the entire tree of life, which we've just realized is much bigger and goes well beyond ourselves. Having the set of tools to change that much more rapidly and much more directly, I think there's no way to predict what that looks like in even 50 years.
So we're grabbing the steering wheel of evolution effectively. So, Eric, any?
Yeah, I think Jenny covered it well, in that what CRISPR-Cas9 has done is it's elevated this into the intellectual discussion of more common people, I think. It's obviously taken science by fire, but even from some of the questions we're getting tonight, it's starting to have a societal impact. And I think that's a good thing. There's still a lot of unanswered questions, but it's a very powerful technology. Has the space of gene editing been really revolutionized? I don't think so. We had the tools before to do these things. It's just been brought a little bit more broadly to the communities to use now. So I think that will push this forward, and that's a good thing.
Yeah. Ed, any comments on?
Yeah, I mean, I'd say that what you've heard about is from a research perspective, an incredible revolution, because CRISPRs make the kind of science that can be done at a research level ubiquitous and available. I think ultimately where I think the greatest impact is going to be is actually being able to move to permanent curative outcomes, one-time treatment, permanent cures. And that can change the lives of many, many, many patients. But the metaphor one can use is like nuclear power, right? Nuclear power and nuclear fusion can be used for good, it can be used for evil. And this is very, very powerful science that can be used for lots and lots and lots of important, valuable benefits to mankind, but it can easily be used in other ways as well.
Yeah. Riley?
I think that, as we said, this is something you can do, right? You can go and you can edit the human germline. You can go and edit cell lines in patients or not. Now the question is, what do you do with this? How do you actually benefit society? And I think that still is very much an open question. I think that we have this really great hammer, and we're looking around for nails. And I think biotech and CRISPR and genome editing, it's still kind of looking for some of its killer apps. What are those great things that will affect everyone's life? If you think about it, biotech has not had as much impact on your life over the last 40 years as semiconductors, nowhere even close, right? Smartphones have changed how we live our lives dramatically. And biotech, unless you are a patient of one of these sort of key areas, you don't even know it's there sometimes. And so I think it's really going to be interesting. When does this go mainstream?
And I'll just sort of summarize. I think that from my perspective, this is a very momentous time on this planet. After 4.5 billion years here, all of a sudden, a single individual creature on the planet is taking control of the destiny of evolution. And that is a big deal. And we don't know where it goes. Now, simultaneously, there are other guys in the AI departments trying to make artificial intelligences that are going to be much smarter than us someday in probably this century. So it's fascinating that these two things are happening at about the same time. Anyway, I want to thank everyone for coming. And Vivek, or do you want to say something in close? Yes. Okay, go for it.
So give a big round of applause to panelists.